Hello, good evening, everyone. We have Todd here tonight. Let me just put down the volume in order not to have echo. Okay, it has not started yet on my other window. Now, let me put it down. Okay.
You're live on my screen.
No, I have just put down the volume, so we have no echo.
Okay.
So Todd, let me quickly introduce you. We have met us at the Creation Facebook group, and I posted there a post about the irreducible complexity of the eye, and you said that you would like to have a chat on that on my channel. So I did set it up, and I would like to know from you. You said that you disagree with my interpretation that the muscles around the eye are irreducibly complex, the system.
Okay, I can share something. First, what would you like me to refer to you as?
Oh, Tangelo.
Okay, I'm Tangelo, because your other screen name said something else. Also, do you have a camera by any chance? If not, that's fine. It's just a lot more personal when I can talk to somebody rather than a black screen.
Yeah,
because I do meetings and people do black screens, and it's just a little more personal.
I can put the camera on, but my wife has used the light.
Okay, that's fine.
So I can put it on, but I think.
Yeah, like I said, I'm not going to make a big deal out of it, but it just kind of makes it a little more personal too. So it makes it kind of nice.
I can connect the cam, but it will be rather dark, but we can try. Okay, just a minute.
It's already dark with the black screen, so even if it's dark, that works. But then, yeah, I'll get going and I'll share some thoughts on that. If it makes you feel any better, I have this light over top of my head, and it's kind of glaring on my eyes, and it's probably making the bald spots on top of my head show up a little bit better. So dark room would probably be preferred at this point. There, okay. That's all personal. Yeah. No, that's not too dark at all.
All right.
Perfect.
Yeah, I did put the light of the cell phone on, so.
Well, so I do a lot of Zoom meetings, not on my channel, but I do Zoom meetings on the side, and I talk to a lot of black screens, and it's just a lot less personal, and then it messes with the brain after a while. There's just a lot more, but again, if there's a bandwidth issue or whatnot, it's not a big deal. So anyways, okay. I can go ahead and start. Yeah, my name is Todd of the OutSci'Ed channel. So first of all, OutSci'Ed channel is kind of something I've been doing for a while. I've added videos, I've deleted them. Many years ago, I posted some interesting things. Then I posted some firearm videos that really took off because it's kind of a hobby of mine. But at the same time, my real passion is in science. My background is in biology. I have field work doing expeditions in geology, also marine biology. But my passion is all of the sciences. So whether it's astronomy or anything else, that's really what I enjoy doing for the most part. Education is something that I'm really passionate about too, because I like working with people of all ages. And I might say for the most part that, maybe I was a little naive starting out many years ago, because I did have some ideas that you could always convince people by just showing them enough evidence, and then people would come around and you could always just show people enough evidence. But over the years, I started to see that this wasn't the pattern. You could show people things, and I'm not talking evolution here. I'm talking just about anything, even politics, and you could even have something, even if it was DNA evidence, in terms of showing somebody had committed a crime and people were still in denial. For example, the O.J. Simpson trial. I can use that for many things, but even his best friend, who was Rob Kardashian, the Kardashian sisters' father, he was at the time very much in turmoil about the DNA evidence, but he still continued to defend his friend, and ultimately he got off. However, after the trial, Rob Kardashian never really hung out with O.J. Simpson afterwards because he had thought about it too much, but yet he still continued to proceed through the trial defending his friend. And that kind of goes along with psychological terms I've since heard of then, which is cognitive dissonance, where people can have two conflicting ideas and they still tend to go with the one which reflects their upbringing or their echo chamber, which is the people that they associate themselves with. And, I think for that most part, I guess I've gotten to the point where I don't blame people if they don't change their mind. It more bothers me if they pursue influencing other people and they don't try to ask questions about these. So I don't anticipate changing yours or anybody else's mind, but at the same time, I would hope that if you have questions, you could ask them, and that maybe at the very least, maybe get a little idea out there that perhaps there's a little bit more to this than I originally thought. Because I'll also give a little introduction too, that my background was creation. I was an ardent creationist. I was with Kent Hovind the month before he went to prison in 2006. I was up on the stage defending him. In fact, I was using the very arguments for irreducible complexity. One of the things I actually threw at Kent Hovind at the time was a softball question. And that was, it goes back to, he had his Hovind theory. Are you familiar with Kent Hovind, by the way?
Yes.
Just check. Okay, so he had his idea, which he called the Hovind theory, and he pushed it. And he also kind of talked about Walt Brown. So to be fair to him, he was offering two ideas on how the Ice Age got created. But he said mammoths are found with food just completely undigested in their stomach. Therefore, they must have been frozen in an instant. He claims he went to the Denver National Ice Core lab, and they said that in order to freeze an animal that large, it must have been negative 300 and some degrees. Kent Hovind said the only thing that is that cold that can do it to that many mammoths, it must have been a flash frozen event. And that was a cool idea. However, I also believed, even at the time as a creationist, you should not be in an echo chamber. Hearing your ideas from the same people, you should hear it from outside sources, and always keep your mind open. So what I did was on stage, I thought I'd throw a softball question to Kent Hovind. I remember just because he was getting hammered by this local teacher and his students that came forward. And I said, Dr. Hovind, just to kind of throw this out there, I said that I did look up some other explanations, including from Answers in Genesis, and they actually were against this idea. And they said that the reason mammoths have food undigested in their stomach is because they actually have a storage pouch. And that is, sure enough, the pouch that the food was found in. And I said that fully explains why it was not frozen. I thought he had an answer, and I really was trying to help him out with the students because I loved the guy. I really did. Instead, I was rather shocked by his response. He instead started telling people in the audience that he hemmed and hawed, and then he kind of said to himself that, well, that's why I call it the Hovind theory. I can take the blame for it, and it's just a theory. And he said, that's why I call it that. So then I can't really get taken the blame for it. And I remember just thinking, like, you're supposed to be the scientist. You're supposed to change your mind or throw out a bad idea if new evidence arises. Now, over time, I will say I really decided that my relationship with God in that too, like I was going to follow wherever the evidence took me. Now, also, I will make a claim that my reason for being here is not to talk about religion, but to talk about science. And, I can venture into religion too a little if you want, but I do want to focus on the science because that's ultimately what we can test. So I will say, kind of just to kind of summarize this, that my background is really about getting to the understanding. And really, to be a scientist, where I've since kind of followed, you have to follow the evidence wherever it takes you. I'll throw this out there too, that I grew up with Jurassic Park. I was in love with Jurassic Park. I loved the image of them, drawing the pictures, and my son is now doing the same thing. He's obsessed with all these dinosaurs. Unfortunately, the evidence has come in that they were, for the most part, feathered. This is not even really a contradiction. Even creationists can enjoy this new knowledge that we've learned. We've actually learned so many things in 20 years about them being feathered. But I got to be honest, I'm putting my feet up. I want those dinosaurs to be feathered. I really do. But I also have to step back and say, I also want to be a scientist. I have to follow the evidence where it leads me. And that means the dinosaurs did not look like the theropods, at least, did not look like the cool reptiles that were portrayed in Jurassic Park. And that's hard for me. I had to break away from that. But that is part of that journey of stepping away from creationism. So I say to you, I am willing to follow the evidence where it leads. And that's actually why I'm interested in engaging in these discussions. The question is, are you willing to look at evidence and change your mind wherever it takes you? Now, whatever we look at here, including the explanations of the eye, that has nothing really to do with your belief in God. There are some things, I admit, that do seem to contradict Genesis, but there are plenty of scientists out there that still are able to kind of harmonize that. Now, I will pretty much leave religion at this, that I was not able to harmonize this over time, and it was a big struggle. And I went to different churches even, hoping to get the answer. And I've been talking because I want to get to your eye thing that you had asked me. But I will say that in 2014, I went to my church when they had the Ken Ham and Bill Nye debate. If you remember watching that in February 4th, 2014, I, at that point, was kind of at a crossroad where I knew there were so many things that creationists were skipping over, making simple. And, you know, we needed to have more dialogue in the Christian community. We needed to have these actual discussions because the answers were not as simple as a Facebook meme. So I went to the church I had been going to for about 6 months because the other church we had been going to, it was kind of the same ordeal. Things were oversimplified. And it wasn't even that. It was also like how they dealt with people in the church. And so we were at this mega church that we were at. We watched it in the auditorium, and I thought this is going to be awesome. At the end of this debate, people are going to say, let's have a discussion. And there's going to be some of us in the audience, maybe with an old creation aspect. Some people maybe that are theistic evolutionists. And then, of course, we'll have Young-Earth Creationists. And we can all have a friendly discussion at the end of the Bill Nye Ken Ham debate. And I remember while the debate was taking place, I kept looking at the people around me. Every single time Ken Ham spoke, the woman in front of me, her body language, she was relaxed. She put her arms down. And she then, every time Bill Nye spoke, she crossed her arms, eyebrows were down, and she just... That was, to me, the body language of somebody that was putting up, as we call it, the God glasses. She was not, and that isn't even really about God, but it was, I am not changing because if anything is a threat, I will not look into it. And that was what happened. Well, at the end of the debate, I thought, well, she's just one person. What ended up happening was the pastor went up front and said, wasn't that a wonderful debate Ken Ham did? Let's all pray for Bill Nye that we save his soul. And everybody, I swear to you, almost 1,000 people started praying, got up, hugged each other, and I was in shock. That is the definition of an echo chamber and no discussion. All right, having said that, your question on Facebook, you were talking about irreducible complexity of the eye. There is, and there's probably not really a point in going through every single little aspect, but I will summarize it, and you clearly understand that, at least from your post. The eye is a very complicated thing. The muscles that position it, you've got muscles that literally just look up and to the left and up and to the right. And then you have the other muscles that allow you to look down and rotate in different directions. You've got the lens that is very good at focusing, well, when it doesn't have problems. You have the muscles within the eye, the ciliary muscles that can tug and focus the eyeball. And of course, it has to focus perfectly on that retina. And I'm probably skipping over quite a few things because I'm making it sound simplified. So the reason that it fails with irreducible complexity, well, I should say, first of all, you're right. If you take one of those out, the eyeball does not work. That is correct. I will be honest about that. But the eyeball does not work in its form fit for you and I. However, there are people out there with cataracts where their lens is covered. Would those people rather have their eye gouged out because they cannot make out the object in front of them? Or are they still better off because they can still tell light and dark, even if their eye is covered? The same goes with many animals or some other creatures. If they have any type of light detecting pigment, even if it can't make out an object, anything is an advantage. And if we pile on to that, that is what we call emergent properties. It doesn't really matter to these creatures. They don't have to have the best eye in the world, but in their niche, in their ecosystem, if it works for them, that's all they need to have. And so if you recall the Dover panda trial of 2005, when they were talking about the bacterial flagellum in the motor. The day that the plaintiffs came in, they wore mouse traps on their ties. And the judge, because the creation side was trying, well, I should say the Discovery Institute, the ID crowd, they were trying to explain why the bacterial motor is irreducibly complex and how it could not have evolved from little incremental changes. And so they gave a very good presentation, but the mouse trap on the tie was a very big sign. And the judge was saying, why are you guys wearing a mouse trap as a tie? And they were saying, well, the mousetrap is irreducibly complex. It has 5 parts to it, including the lever that holds it back, the spring, the main bar, the trap lever. As a mousetrap, if you take any one of those parts out, the entire mousetrap does not work. We'll take out 3 parts. All you have now is a spring and the main lever. And then what they did was they said, it does not work as a mousetrap anymore. This thing is a complete failure. However, it now has a function as a tie clip. And it does not work as a mousetrap, but it still has a function. And in biological systems, anything that has an advantage over not having something is a gene that is likely to stay around by natural selection. So same thing with the eye. It does not have to have a perfect focusing lens. If an organism has a pigment or something on its body that's light detecting, it doesn't have to be a perfect focus, but it will give it some advantage to us. But you're right, in a human eye, anything we take out, it's not going to have its original function. That's not what biologists are trying to say. So I've been talking a while and I apologize, but I had to introduce and then go ahead and throw that in there because I was asked to answer the question before I got to introduce or anything. Go ahead, sorry.
So what is the main protein which is responsible to receive photons?
I'm not familiar particularly with the protein, but is it rhodopsin are you talking about?
Yes.
The pigment rhodopsin. I'm not too familiar with it. Okay.
Okay. I'll put a slide here. You can have a look. Just a moment. Okay. So you have here the rhodopsin, you have the retinal, and the opsin,
Okay.
7 transmembranes. Which one evolved first, the retinal or the opsin?
Well, I have to kind of stare at the slide and take it all in a minute to fully understand how to, how I would answer the question. But I'm not a biochemist and I wouldn't proclaim to be one. And so I don't know. And, as a scientist, I'm okay saying that. But I will make a prediction here, and this is what evolution, and this would be my answer. Evolution would offer a testable prediction that any one of those functional groups or any part of the precursor molecule would have an advantage, maybe not light detection or maybe not photoreception, but it would have some other advantage that would allow that creature to better survive in its environment. But it's really pointless for me to try to give a biochemical response to a slide like that. But that's the best, most honest answer I can give you at the moment.
Okay. Science has no answer to that question.
Have you consulted a biochemist who has researched that topic? Because
this was published in Nature, and they were right. The problem here is that even as far back as the prokaryotes, the complex 7 transmembrane domain arrangement of opsin molecules seems to prevail without simpler photoreceptors existing concurrently. Darwin's original puzzle over ocular evolution seems still to be with us, but now on a molecular level. So whenever we try to give an explanation in regards of origins and of biological complexity, we have to check in biochemistry at that level. It is meaningless to look at fossils and transitional fossils, because the action where actually the mechanisms lie which build complex organisms is on the biomolecular level. So what we have here is the rhodopsin protein, which is made of 2 parts. That is the 7 transmembrane, which there are no precursors known. So the question would be, how did that the opsin evolve? And it is very complex. You can see here what they are writing. The more that they studied it, they say that the increased complexity was stunning. Knowledge of protein folding is important because proteins must assume the correct 3D structure to function properly. So you need the correct 7 transmembrane opsin protein. in order to have function, but not alone that. You need also the retinal, which is that structure here. And they say all structural details in the retinal chromophore are functionally important. That means if you don't have it in exactly that form, it will not be functional. But there is more on it. This is a very complex interplay between the opsin and the retinal. And here it is described, okay? The binding of the chromophore to the opsin is essential to trigger the conformational change, which in sequence triggers the signal transduction pathway, which transmits a signal to the brain, ion channels for flagella beating, etc., and must be precise and functional from the beginning, and following is required. A shift-based linkage, a lysine 296 residue where retinal covalently binds, it must be exactly bind at that residue, and it has to be a lysine residue, then the side chain of the residue, and an essential amino acid residue called counter ion, which is essential for rhodopsin to receive visible light.
So if you had a question, go ahead, sorry.
So what this means is that this is a system which has to be right just from the beginning, and in special also retinal, as I told you before. And they say here that all structural details in the retinal chromophore are functionally important. So rhodopsin doesn't work if both parts are not in place. And the 5 points, as I described, they must be exactly right in order when a photon is hitting the retinal, it will perform a conformational change in the pocket. The pocket also has to be exactly right, and then the signal transduction can be performed, okay? So this is a system which, in my understanding, is irreducible, and it has to be very precise, and retinal by itself has no function, and opsin has eventually other functions, but the system only works together.
Okay, so your argument again is going back to irreducible complexity. Now, this is kind of cut and paste science where it's in front of you here. I'm not sure if your background is in biochemistry or not, but again, I see something here that is rather complicated, just as I was saying earlier. And you were saying irreducible complexity means if you take one part out of it, it will not work. But yes, again, I have to say, my background is not a biochemist. I'm not going to pretend I am, but I can say, looking at this, it looks very complicated. But are there more simple monomers it can break into where they have other functions that are not retinal-based? Could they be a derivative of metabolism? Could they have been a waste product that was up in the head? I don't know. Could they have been adapted for something? I will say these are good questions. This is the scientific method. And so you're doing a good job with that by asking these questions. In science, what we want is to have questions because it gives us something to work for, because we don't have all the answers, and that's okay. In the scientific method, you have an observation. This is very complicated. Now you're asking a question, if you remember the second step of the scientific method. Why is this so complicated? So you come up with a hypothesis. You are saying it is so complicated, it is used as retina, and it cannot be derived as anything else, okay, or it won't have a function. But are there other hypotheses out there? That I don't know, and I'm not going to pretend to. But we as scientists, we do not want to skip the next crucial step, which is peer review. Because even if you did come up with the idea that it was irreducibly complicated and nobody could disprove that, it would be a potential theory. And it potentially, let's just be honest, it could debunk much of what we know about the evolution of the eye. But like all things in science, nothing is ever proven because a theory is tentative, even cell theory, even the theory of evolution, because nothing truly can be proven. That's why you've heard the saying, I think, therefore I am. Because Rene Descartes' idea of his, Cartesian reduction, the idea is the only thing we can prove with 100% certainty is that you exist. Everything else is probability. Even if we do get nobody to prove this wrong, that it is irreducible complexity, all we are left with was a theory with, well, good evidence behind it. I am not seeing the evidence behind this. I'm seeing something presented in front of me that is complicated, and I'm being asked to respond to a biochemical pathway, which is not my background. But I do respect this. I think it's worthy of looking into, and I say, commend you. You've done the observation. It's complex. you're asking questions, and you have a hypothesis. So now you need to test this out, because here's a hypothesis that I have. Could you look at the derivatives of this molecule, and could those have another function? Could they have been created from another metabolic pathway? And that is something, unfortunately, I don't have the moment or the resources to test. But I would say as a scientist, I'm okay with that, because we do not have all the answers. But that is a good thought to bring up, though, anyways.
Okay, I'm a machine designer. That's my profession. So when I started to investigate and learn about biochemistry, I saw parallels between what I do as a machine designer and what I see happens in the cell, like the genome, which directs the making of proteins and controls the operation of the cell and so forth. So that has led me to understand why this is a problem for evolutionary biology. Because, for example, as I said before, the retinal, it has to have a correct size, a precise structure in order to be functional. So the question is, how do you get that structure? And on the right side, you see the biosynthesis pathway to make a retinal. And the question is, if you have only retinal, but you do not have the opsin and the right-sized pocket of the opsin where it is inserted at the right place at the specific amino acid residue, you do not have function. Now, how do you get to that structure by evolutionary pathway? And let me read what they write here.
That's the hypotheses to be tested by a biochemist. I don't have the background in that. But I will ask you this, because you said it poses a challenge for evolutionary biology. If you were walking down the road and you saw a ball fly upward, would that be a major challenge to the theory of gravity? And we throw out all of Isaac Newton and everything we know of Albert Einstein. What would your answer be to that?
Well, I would like to make a parallel to an engine. When you see, for example, a car motor, you have a cylinder and you have the piston. You agree with me that the piston and the cylinder, they need to have both the precise size in order to be working together. If the cylinder has a too big size or the piston is too long or something like that, they will not interact in a correct manner together. Do you agree with that?
Again, I just so we're not like posting a lot of complex things and trying to win by default by, going through and I'm not trying to make this, oh, shoot. Sorry, give me a second. I'm getting a message on low disk space here. Give me just a second. I'm going to delete some files here. There we go. I'd hate to be cut off by that. Okay. But again, I have to ask, if you saw a ball fly upward, would that disprove all of Albert Einstein and Newton's theories on gravity just by seeing a ball fly upward on a sidewalk?
Well, Todd, I think that we can draw analogies to what we see doing.
Again, please, just if you saw a ball fly upward, would it disprove all of the theory of gravity? Just that one observation you make, just out of curiosity. It wouldn't. Thank you for responding, though. But it could be a challenge. And I would say you're right. It could be a challenge to the theory of gravity. But that's where we have to we have observed something. We have to ask a question. How the heck did that happen? We now make a hypothesis. We would consult people in the field. It could be something really stupid, that there's a string. There's an alien shooting a tractor beam down to pick it up. There could be a fan, like a vent or something, that Marilyn Monroe stood on with her dress blown upward. It could be on the sidewalk, blowing it up. We wouldn't automatically just say, well, that disproves the theory of gravity, and then go and start sharing stuff on its complexity. We would say it poses a credible challenge to the theory of gravity. We would instead say this is worthy of research in there. And so I wouldn't know to say if this poses a formidable challenge or not. And if anything, it might even if this was valid, it might pose a challenge to a certain aspect of the eye. But to then say all the other evidence that we have, just toss it out. Again, that's where I'm just saying, be careful with how you say that and your meanings behind that.
Well, I can give you many, many different examples where I think that...
But each one of them, it sounds like they're coming from the aspect of irreducible complexity. And again, the argument is that if you remove one part, it will not work. But again, what the scientists are telling us and what we can observe in the anatomical structures of animals is they can have simpler forms of this, but they might not have the same function in them. But as long as they convey an advantage, that's all we really have to do with it. So,
well, I came to the rhodopsin thought because this is the protein which is key. If you do not have rhodopsins, you do not have the molecule which receives the photons and then starts the signal transduction pathway. So this is, you cannot go more simple than that or lower than that. This is the key protein. So that's why I brought it up.
I will concede I am not a biochemist, so I don't think it's worth probably going into this more on the audience's time. But I will say that a biochemist would be worthy of looking into this, testing out that hypothesis. My prediction on this is that they will find that there's something else it could be used for. Or if it can't be used, it was a waste product. And I'll give you an example of that. The air that we breathe right now, oxygen. Oxygen has no useful function, believe it or not, if you are an anaerobic organism, because oxygen is actually a waste product created by photosynthesis. It's used to create, not to get complicated or anything for the audience, but it's used to create a proton gradient where hydrogen. What they do is it takes water and they break off a hydrogen, and the plant can use the hydrogen to run photosynthesis and make ATP. The oxygen gas is a waste product and it's toxic. It's an oxidizer. And so the plants get rid of it. And billions of years ago, they did the exact same thing. However, there was life that found a way to use this waste product. And they used this waste product because they were able to use it and oxidize the products of the electron transport chain. Oxygen now doesn't do anything to us. When you breathe it in, all of our electrons in the mitochondria, they pile up on it, and the kind of like, I don't know, maybe a burglar or something going into your house and stealing everything, oxygen then flies away with all of those electrons. That's all it does. When you and I breathe oxygen, it is a waste product, and it can hurt us too, by the way, in large amounts. All it does is carries away the electrons, and it makes our body much more efficient. And that's kind of the same thing. Like, oxygen did not have a function, but our body found a way to use this waste product. So I would say evolution can make that prediction that it either has a function in a primitive form, or it was a metabolic product that the body found a way to use another purpose for. So, and again, I would ask you if you have other examples or you would like to get into another discussion, but if you're just putting up biochemical pictures and stuff, I'm not a biochemist. I'm not going to be able to respond. And I don't think that's fair to the audience either, to just show them complicated pictures and try to win by overwhelming.
No, it's I try to make it as simple as possible in order that you and the audience understand it. So do you know what
rings, multiple pathways?
So do you know in photosynthesis which part produces oxygen?
Yeah, the photolysis, photosystem 2.
Yes, but it is the oxygen evolving complex.
Okay.
The oxygen evolving complex, it has 5 proteins. Let me check here. Okay, it has 5 proteins. Here, it's PSPO, PSPQ, PSPR. In total, there are, as far as I know, 4 proteins. Okay, so you can see this in blue here. They made knockout experiments, and they did find out if either one or the other of these 4 or 5 proteins is missing, then the oxygen evolving complex does not work. So
do you also know that photosystem 1 does not do photolysis, and that is also used by some cyanobacteria? So photosynthesis does not require oxygen either. That actually evolved later.
No, we are talking about oxygenic photosynthesis.
Sure, but that conveys an advantage, but it wasn't necessary. Just to make that clear, it wasn't necessary for photosynthesis.
Okay, but it is necessary.
Oxygen was kind of a benefit because it gave more hydrogen to the plant, but they did photosynthesis before they did photosystem 2. It's more efficient, but it wasn't necessary.
Okay,
you can burn a campfire with firewood, but if you blow oxygen on it, it's more efficient. That's what I was trying to say.
Okay, they are described here. 1, 2, 3, for 5 proteins which are surrounding a molybdenum cofactor in the middle. It is pictured here. Okay. And water splitting is a process which is performed in 1, 2, 3, 4, 5 steps. Okay. And the system, they made knockout experiments with each of these proteins which are surrounding the cofactor, and they have proven by these knockout experiments that all 5 proteins, they are absolutely essential. And I'm showing that here, okay? The photosystem 2 manganese stabilizing protein PSBO is known to be the essential PS2 extrinsic subunit. And they made it with all the 5 proteins. And these proteins, they have only use in the oxygen evolving complex. They are not used in any other biochemical system. And the cofactor, which uses molybdenum, is a very, very complex structure, and water is also absolutely essential in that process. So this is another example of irreducible complexity, where if you
I still have to go back. I'm sorry, I still have to go back just because we're at almost 40 minutes into this now. And I still have to go back and say that the main argument is irreducible complexity by trying to throw biochemistry, which is not my topic, into this. In fact, very few people even probably watching this have that field. And so even if I did have a good discussion with you on this, that would be hard, you know, it would be very hard to give an appropriate response, and I just wouldn't want to make that misconception. But I'm not saying that there isn't a good response. Theoretically, or not even theoretically, it is very possible that irreducible complexity is a valid hypothesis. However, every time it has been brought through peer review, that has failed. It has not shown up. So here we are in a discussion with people that don't have a biochemistry as their predominant background.
Yeah, let me interrupt you, because these papers,
we're not beating around this too much. That irreducible complexity, the reason it fails every time it has been brought into peer review, and it was a valid hypothesis 20 years ago, by the way, it was. But the reason it always fails is because there are precursors to these more complex emerged molecules. And each of them either had a function or they were a waste product or something like that. And later they were able to adapt. That was the biggest thing. And the same thing with the bacterial flagellar motor. The same argument Michael Behe had thrown in there in his book in 1995, Darwin's Black Box, where he said, much of his book, look at how complicated the bacterial flagella is. He even quoted scientists as saying that it appears that it was designed. It is so complicated because that motor that spins the flagella tail, you know, like on a sperm, it is so complicated that it cannot be explained, because if you take one piece out, it will not function. The problem is the scientist he is quoting in the book, Michael Behe, he later went and he actually had a part in the trial where he went and said, I did say it appears that it was designed, the bacterial flagellar motor, and he was actually one of the most renowned scientists in the world on the bacterial flagellum. He said, I did say it appeared designed, but I didn't mean it did, or it was designed. I'm just saying it looked designed. He then went and showed part of his research that was on a kind of injection needle, if you will, on a similar type of bacteria. This needle can be used to inject poison. It is on every bit of way the same as the bacterial flagellum, the same proteins, and the motor proteins used to spin. There's only a couple pieces out, but they are still there. And even though it was not a spinning motor, it had another function, and that was it served to inject poison. This was a toxic type of bacteria that would grab onto you with its pili, and it would use its injector. They have a proper name for it, proboscis or something, and it would inject you with poison. And that, again, is where I would say, if this is all we're going to do, it's going to be my same response. I'm not a biochemist, and it's not fair, if there is a biochemist watching, for me to pretend that I have answers. But I can say that is the failure with irreducible complexity. And I will also say, because I don't know your background, every time I talk to a creationist about this, with less complicated things, by the way, than biochemistry, and more things that were on anatomical structures, it was still, and this was when I was a creationist, I would talk about, well, what about, this is what the evolutionists would say. And the argument was always to deflect or ignore. And there again is that term cognitive dissonance, when you have two conflicting ideas and you stick to the one that helps you sleep at night. And that's unfortunate, but that is our infallible brain as a human. And that, again, I would also kind of throw out there, shows flaws with our brain, because our brains are capable of being irrational like that. They're good enough for us to survive in the African savannah thousands of years ago, but they have flaws today, and that is reasoning and logic. That's just kind of where it is. So, I mean, I can throw up some things here. You wanted to talk about origins, and like I said, we're 40 minutes in, but it's still the same argument of irreducible complexity. And let's be honest, irreducible complexity, it's basically saying, I don't know how it happened, therefore God did it. It's that argument, God did it. And that could be a valid hypothesis. And in the creation group, a lot of people are trying to say, why do people say God couldn't do it? Why do evolutionists, and they say atheists too, by the way, because atheists, evolutionists are not synonymous, and I really like how that's always thrown out there. They always say, why do atheists or evolutionists always say that God couldn't do it? And I don't know how many times I have to say this. No scientist, well, no good scientist would say that it is impossible for God to have created anything. God could have done it, but there is no evidence cited behind this. There's no testable hypothesis. For example, if a body part or something, or a part in the cell was formed by God, this is our hypothesis, God did it, how could we make an experiment to test for that effect? What would the experiment predict that we could look for? So in one example I can give you, 10 years ago, I read this, it was Frank Tipler's book, I think his name was, and he was talking about the physics of Christianity. And I was fascinated because I, again, was Young-Earth Creationist, and I was reading this book by Frank Tipler. And he was talking about Jesus when he was buried and when he was buried and whatnot, and how he rose from the grave. And he was trying to use physics to describe how Jesus could have come back to, or how he could have rose from the dead. And of course, as a Christian, I was like,
sorry, let me interrupt you. Before you continue with another topic, I would like to respond to the example which you brought forward with the flagellum, okay? And then you can continue your narrative. Okay. I have made an investigation, and the flagellum uses 36 different proteins, okay? I have researched each individual protein, and you can see on the screen that what is written in red. Science confirms that each of those 36 proteins is irreducible. If you remove it, you lose the function of the flagellum. And I can provide you afterwards the link, and you can look it up. And this is all peer-reviewed science. It is not something which I am bringing up from creationist websites or something. It's my investigation.
And you're right. If you do remove it, it doesn't function. But did you not hear what I was just saying about it had another function, and that was the scientists were saying it was an injector.
This is a simplistic explanation. First of all,
not simplistic. There still are organisms with these injectors on them. They have the exact same structure.
Let me explain to you. To go from that molecule to the flagellum, you have a difference of about 40 different proteins which you have to add in an orderly fashion, and and you need also assembly proteins to make the flagellum to build it, and these are different proteins. Okay. So this is the same. I like to make analogies because it is the same that we require. I want to give another example. For example,
to answer that question real quick, you were saying, and that is in all fairness a question, so I'll answer it. You're saying, because the one thing they were saying, the injector that was injecting poison, you're saying, well, even from there, it's 40 proteins. Well, I don't remember that being what they said, but let's just assume it is 40 proteins. And they would have to be in the perfect order in order for it to be this flagellum. Okay, that is a fair argument. But again, I don't know a single biologist, and I'm really saying this, especially if there's other people watching, this is not chance. A lot of people mistakenly say that evolutionists believe that it is all chance. That is not how evolution or natural selection works. Because again, you can have 2 amino acids fall into place, and they could have been just a mutant and sitting there. And in that sense, that little part could have been chance. But again, we're just talking 1 or 2 amino acids. However, if you get 3 or 4 in order, if they've conveyed an advantage in any way, perhaps that injector was more durable. Perhaps the injector had a larger pipeline motor on the side that allowed it to put out more poison. And again, I would hate to give too much of an explanation and just be accused of not knowing biochemistry, because I've said over and over, I am not a biochemist. But today, it has 40 different amino acids in those orders. But what did it look like in the past?
40 proteins.
Each of those proteins, and again, this is evolution, it's divergence. Did each one of those proteins that got added, they would have had some other advantage to it. Now, it might not have caused the spinning.
This is speculation. This is speculation, because if you that doesn't work. You have the flagellum only if you have all
science. That's the beauty of science. We get to ask these questions. We get to go and take a look at it.
It has been explained. I am showing you that each of the,
we don't know why this happened, therefore God did it. That's not science.
I am showing you that each of the 36 proteins used in the flagellum, they are indispensable. If you have 33 proteins, the flagellum will not work.
So the Discovery Institute, if they're watching, the Discovery Institute had that chance. They wanted their court case in 2005, and they got it. They got it with a highly conservative judge, too. And in fact, it sent the National Center for Science Education running, because they were terrified. We have this George Bush appointed, very religious, right-wing conservative. He's presiding over the case in Dover, Pennsylvania. And they were terrified that the Discovery Institute was going to actually implement intelligent design in science classrooms. And what ended up happening as a result from all this result was, when the Discovery Institute had their chance to go up, a bunch of their witnesses did not show up. They backed out last minute. Michael Behe went up there, and he made it sound just like you are. Very complicated. All these have to be in order. And that, by the way, was when the cross-examination did the whole case of irreducible complexity in the necktie. But then they went in and started asking Michael Behe about his ideas on what science was and about its ability to be tested. And instead, Michael Behe, through cross-examination, he came out and said what I thought was just baffling. He said even astrology is science. Astrology. That is not science. And how a man who works at, I think it's Lehigh University or whatnot, has that idea of science, that is baffling. Astrology is not something we can test, and when it does, it fails. And at that point, the Discovery Institute looked ridiculous. They had their day in court, and so they completely failed. Michael Behe presented all of this argument. And so again, I'm not going to try to sit and waste audience time and pretending I'm going to come up with explanations. That is the role of a biochemist. They had their chance, and Michael Behe in his book laid out his case, and Judge Jones of that case was so upset. The Discovery Institute and all of them, I'm not the Discovery Institute, but the Dover School Board who brought in the discovery, they had to pay the court costs because the case was so ridiculous when put under cross-examination. That is what happens when you actually have people putting out this material under cross-examination. Instead, where do creationists thrive? And this isn't, an insult or anything, but they thrive in the church setting, in their echo chambers, or on the internet message boards. They do not thrive in peer review. And I know there's a lot of people that say that their materials just don't get published. No, it just doesn't work. There's a reason it doesn't get published, because when scientists disagree with it, maybe there's something wrong with that. Maybe there's something wrong with the hypotheses. And again, I want dinosaurs to not have feathers. But as a scientist, well, I'm not a scientist, but as a person of science who's fascinated by this, I have to follow the evidence. And you need to follow it too. So look up the Dover Panda case, and random people on the internet trying to go through this, it's not going to work. I have something for you since I've been just responding to claims.
I would like to respond to everything that you said right now. Okay,
because then I would like to present some things too, because I've been
I think we have here about you talking about 80%, and you are not listening to what I am presenting. So I would like that.
I would wonder what side that audience is too. Again, that goes back to echo chamber. Look up the definition of echo chamber.
Well, echo chamber, you are only talking, Todd, but you are not listening to what I am saying.
We've been 15 minutes on irreducible complexity. I would like to show some case stuff too. The idea was on origins.
That was the topic that you brought up when I brought up.
I haven't even talked about origins yet, sir. I've literally just been trying to give an answer on irreducible complexity. That's why it doesn't work.
I brought you 3 examples of irreducible complexity. You have not refuted it. You just say,
I'm not a biochemist. Why would I refute it?
So you are,
I'm not
yes,
a biochemist.
yes, but you said that you would come to my channel tonight because you were
I didn't know
opposing.
what you were going to talk about with the specific stuff. You're talking metabolic pathways. This is honestly just sounds like a gotcha. I mean, you didn't present the specific topics we were going to talk about here.
Well,
So
I started
if
with
you
the
would
topic that
respond
you were
to, I would ask you to
I
respond.
started with the
I'm going to
topic.
be quiet here. I promise. If you could respond, please, to the idea of irreducible complexity. If you just would like to say, without throwing up another slide and say, look how complicated this biochemical is. If you can do that, go ahead. But then I would like to share something.
Okay. Do you think that the cell requires a minimal size and a minimal number of parts in order to be able to be alive?
Sure, because if a cell is too small, the atoms themselves would be just inside of it. It has to have a minimum size for atomic motion in that. But as far as does it have to have a minimal amount of parts into it? Well, that is what protocells are. And if you're interested in the works of Sydney...
Well, protocells are not alive. We are talking here about the threshold.
Okay, but what does it mean to be alive? The definition is really broad, by the way.
Well,
we have to kind of look at our changing ideas on this too, because at the same time, what life was in the past versus now, we don't have examples of. All we can do is look at what simple cells are. Viruses very much are biological systems, but by our knowledge today, they are not alive. They do not reproduce on their own, but at the same time, they have their own genetic sequences. We can see how they're related to one another, but they don't have a cell membrane. They don't reproduce on their own. They don't eat. So again, we have to have ideas on what are alive based in the past.
Okay, would you agree that reproduction, metabolism, homeostasis, nutrition, complexity, organization, growth and development, information content, hardware, software, entanglement, and permanence and change would be a description of what life is?
Today, and by the way, complexity, that's not something I really have, that's a very vague word I would throw in there too. No, complexity I would not say is a necessary thing to have. You have to have reproduction. And that is, if you want to look up the RNA world hypothesis, depends on what you mean by complexity with that. I would say, yeah, it's complex. It's a biomolecule. But biomolecules are also found on asteroids. Does that mean that they have to have some type of miraculous development to them?
Well, they are not alive, correct?
Well, they're not alive, but again, you're looking at life today and putting on a definition of alive to them. What does that mean to be alive in the past? And again, the virus is around today, but we don't call it alive because we're not comparing it to modern systems. It doesn't reproduce, but it uses a host. But again, what about more simple cells? What do you mean by complexity? That's kind of the thing where it gets a little vague on what all this is.
Well, we can start with the first one. Is reproduction required to have life?
Okay, sure. Because without reproduction, you don't have natural selection, which is how life diversifies.
Okay, in order to have reproduction, you need DNA replication.
No, you don't. No, you do not. Where did you...
So can you give me an example of a life form which doesn't require DNA replication in order to perpetuate itself?
Sure. RNA, I almost said ribosomes because ribosomes are made from RNA. RNA is, as you know, just like DNA, but it's half-stranded. It uses uracil, a few other things different because it has a different sugar. RNA can actually self-replicate. It can. Yes, it can. If you put RNA in a glass of water with other mononucleotides in there, it can replicate. And the reason why is because it's a template where the nucleotide, because if you have a T, RNA, it's a U, A, C, G, etc., the mononucleotides can bond to each corresponding base. And then when that strand breaks off, it serves as a template for another one on there. Now, this is not directed. It's all just, molecular motion. There is no direction on when it reproduces, but it can self-replicate. That was the Nobel Prize for 1989 on the discovery of ribosomes, which is RNA, and their ability to also catalyze.
Okay.
Yes, RNA can reproduce, but it does not have to be in a cell, does not have to. DNA today is what life forms have. Viruses, many of them, as you know, use RNA. They do not reproduce on their own, but again, they don't have to. We're talking about life forms today.
Okay, let me read this. The very origin of the first organisms presents at least an appearance of a paradox because a certain minimum level of complexity is required to make self-replication possible at all. High-fidelity replication requires additional
functionality. Sorry, before you finish this, just curious about the source of this. Is this another creationist website or is this a legit source? And I ask that, not that I'm not going to listen to the information, but sometimes questionable sources will throw things in there that are just completely false, and then I have to second guess myself on everything.
Okay, this is from the book The Logic of Chance from Eugene Konin from 2012.
Okay, I can really wonder, but okay, I just wanted to be fair on that because sometimes there's misrepresentations and then it's like, okay, do you agree with this or not? And then I have to respond to something that might just be kind of rubbish altogether. But okay, go ahead.
Oh, I can give you the source afterwards and you can look it up by yourself. But if you think that Konin is not a trustable or a good source, then I don't know what is, because he is very respected scientist.
And that I'm not saying much about. I'm just, again, when all I can see is writing, and then I have to, I'm being asked to respond to it. But at the same time, there could be things in there that are just flat out wrong. I don't know. But this is one of the, again, common things that are done with creationists is sources that are not reliable. And then you end up kind of being put in a trap like this because you didn't have a chance to fully read it or vet it. So I guess what is the question, or what would you like to get at with this?
Oh, as I said, I can afterwards give you the link and you can look it up and you will see that is from his book. And he says here, the very origin of the first organisms presents at least an appearance of a paradox because a certain minimum level of complexity is required to make self-replication possible at all. High-fidelity replication requires additional functionalities that need even more information to be encoded. The crucial question is how the Darwin eigencycle could have started. How was the minimum complexity that is required to achieve the minimally acceptable replication fidelity attained? In even the simplest modern systems, such as RNA viruses, replication is catalyzed by complex protein polymerases. The replicase itself is produced by translation of the respective messenger RNAs, which is mediated by the immensely complex ribosomal apparatus. Hence, the dramatic paradox of the origin of life is that to attain a minimum complexity required for a biological system to start on the Darwin eigenspiral, a system of far greater complexity appears to be required. How such a system could emerge is a puzzle that defeats conventional evolutionary thinking, all of which is about biological systems moving along the spiral. The solution is bound to the unusual. The origin of life, or to be more precise, the origin of the first replicator systems and the origin of translation remains a huge enigma, and the progress in solving these problems has been very modest. In the case of translation, nearly negligible.
Okay.
So basically that refutes what you tried to claim.
No, it doesn't. Because again, I'm talking about the source here because I'm looking at that and it says basically that there's no way this could have happened without these complicated mechanisms. Perhaps you recall hearing about Thomas Cech or Sidney Altman and their 1989 Nobel Prize on RNA and its ability to catalyze, because we're talking about guys here that started out with RNA and other RNAs. We're talking about chirals that, again, creationists had said for a long time, with all these other chirals out there, left or right-handed, they can't exist together. But when we put them together, they actually not only exist together, but they, in the conditions that were provided in the early Earth, they actually replicated. And not only did these replicate, over 16 generations, they began to increase in complexity, and they started to, in their lengthening, fold up on each other and became ribozymes. Now, Thomas Cech and Sidney Altman, when they did these experiments, they thought there has to be enzymes in there. They did everything they could because at first they were not even interested in these ideas of RNA. They thought RNA was a boring topic. And what they did was they tried everything they could to wash out any proteins which would have been enzymes. All these transcriptase and polymerases you're talking about, we all thought years ago, I always heard about them, they were necessary for a cell to make copies of DNA. We thought this. But their experiments did not show that. After all the trial and error of getting rid of this, all they found was RNA, and not only was it copying itself, it was increasing in complexity. And this RNA, it's also more likely to mutate because, again, RNA is single-stranded. Every base pair is exposed and much more likely to do things. At an hour in, am I able to start sharing some things? Because I'm just kind of refuting ideas here.
Well, I understand you didn't refute anything, Todd, but anyway, go ahead.
But I just said RNA can copy itself, and you're putting up a lot of texts with things that say it can't because it has to be a minimum complexity. 1989, ribozymes, you can look that up. It is RNA can catalyze. Not only can it self-replicate, it can catalyze. Enzymes were not needed in the early days because RNA was able to function as this. These were called ribozymes. This has been known for almost 30 years. And so why are we still talking about this, is my question.
Well, do you know what
because RNA is shared in creationist circles? That's what's going on. It's being churned out in churches, and books are being shared online. But in the open arena of science, this is why this stuff isn't published, because it's been 30 years now we've known about RNA self-replicating. This is okay, kind of why it's hard to even talk about.
Let me ask you, what is RNA composed of?
RNA is composed of mononucleotides that are polymerized.
Okay, what are mononucleotides composed of? RNA mononucleotides.
Okay, carbon-based molecules, nitrogen, hydrogen, the basic things that are in organic chemistry.
Okay, they are composed of, they are composed of three parts: the base, the ribose.
Yes, I'm not sure,
and the phosphate. Okay.
Okay.
So the ribose.
Yes, the sugar, phosphate, and a base.
It can be 6-membered. It can be 4-membered. If it would be 6 or 4-membered or had another composition, it would not work. There was no selection mechanism on prebiotic Earth to have a 5-membered sugar. That's one of the problems. The list which I wrote up.
You understand that?
The list which I wrote up.
That molecules can also form under natural conditions.
Can I finish, please, Todd? The list which I brought up will give you 28 unsolved problems today, not 30 years ago. Today, in order to make RNA molecules in a laboratory, not even on a prebiotic in prebiotic conditions. In a laboratory, they are unable to synthesize RNAs, and it is a huge problem. It is a huge problem. So I have 2 videos on it. You can look it after it.
Right now, there was whatever the name was, their laser experiment where they wanted to simulate the power of an asteroid because, by the way, I'm sure you know about Stanley Miller and Harold Urey and their whole idea that is completely misrepresented by creationists.
That has nothing to do with RNA. That has to do with...
No. I'm just saying about synthesizing abiotic molecules. There's still this argument out there that often they cannot happen.
That's about
later, where they wanted to test the impact of an asteroid. These were all created. Now, again, this is not saying we know exactly what happened in the early Earth because nobody was there. And by the way, well, that's another topic.
It is not nobody was there. It is a concept.
It's not what science has shown us.
Todd, it is a conceptual problem. There was no selection mechanism. You need a 5-membered ribose, which is right-handed. There was no selection mechanism to sort out right-handed from left-handed ribose. Then the bases, there are 4 or 5 bases used in life, which must be just right. They need the right composition of nitrogen and carbon. If you do not have the isomeric composition, then you will not have later on DNA Watson-Crick base pairing. It has to be the right composition. It has to be the right pyrimidines and purines. The phosphate has to be annexed at the 5' position. There is no mechanism known how to bond the 3 parts together. And I can go on. I have listed 28 different problems, and these are unsolved problems by today.
Okay, like I just said, it has been done in a laboratory. You can look this up, where we have artificially simulated these mononucleotides.
Show me the paper.
How they polymerize is another story, but that is a part of research as far as the polymerize.
because I don't know of any paper which confirms that it has been done in the lab. I don't know of any. But
and that's why both of us are not biochemists. We're just, we're not biochemists, so of course would we have access to
I have the internet.
These have formed naturally.
I have not seen any
form naturally. And so this idea that these can't form naturally and then they can't polymerize again goes back to God did it, because I don't know how to explain it. God must have done it. Even if God did it, let's entertain that hypothesis that God did these. If he did this, and again, that could be a possible explanation. But if God did this, what experiment could we do to test this out? There is no experiment you can do that. Please. No, please. Sorry, because then I've changed subjects. There is no experiment you can do to see if we have RNA, did God create these mononucleotides? There's no experiment you could do. So that could be possible, but scientists therefore would have to say God is outside of science. That doesn't mean he didn't do it, but we therefore cannot do this as science. But what we can do is say, let's look at another hypothesis. Could a large impact with enough heat and temperature create a large variety of these? Could they create amino acids, other sugars, lipids? All of these have been tested out. You may have heard of Oparin and Haldane's hypotheses, and every time we get fantastic results with these. That does not prove how a cell starts, I admit, but it does show naturalistic events can create these basic monomers, the building blocks of organic chemistry. Yes, it does. And that's where, back and forth on it, I'm sorry, you're wrong on that.
Well, I will be happy after our chat to send you the link with the list that I have. And if you're able to give me the answers to these open questions, then I will be very happy to acknowledge it that science has found an answer. I have investigated these topics, Todd, and I have not seen answers in regards of these issues. Now, to the thing that you asked, can we test intelligent design? I want to give you an example, and then you will answer me which explanation fits better the evidence. You know that in biology, it is information which drives the mechanistic part in the cell, that is the proteins, which are molecular machines which perform all the work, and they form into metabolic pathways where they produce the products, the basic building blocks, and so forth. So let me suppose that you see a blueprint, and then you see a factory made upon that blueprint, and you have no idea who made both, neither the blueprint nor the factory. And someone asks you, Todd, what do you think? What is the best explanation of both the blueprint and then the factory made upon that blueprint? Is it better explained by intelligent action or by chance and random unguided events?
So this is a classic, the blind watchmaker argument. And in the case of the factory, I wouldn't say it's the same as life. And I can definitely go through that and give other ideas, because biological systems are not the same as a factory, first of all. A factory
is in a literal sense, and that can bring you.
Not the same.
I can give you over 30 science papers which describe the cell in a literal sense as a chemical factory.
Sure, we can call them analogies because the ER, of course, is transporting.
This is not an analogy. I am talking about cells as literal chemical factories, and back it up by many, many different science papers.
I would agree it's a factory too, by the way, but a biological system is self-replicating and changing, and some of those do not work. They get tossed out and others do. Self-replicating is the key to this. The factory is not. So I'll throw this out there. Eric, please, let me just respond to this real quick. Okay. So Eric Hovind, his dad was in prison. Eric Hovind took over and he started running Creation Today. And there's this thing he constantly would throw out there, and it was very much a mockery of this idea that things can get ever more complex and change. And he takes these CDs, and it's like evolutionists say that if we take these CDs and we just throw them on the table, over time they'll evolve copies of Windows 10. And he just throws them on the table and and of course, him and the host just start there giggling and all that stuff. And the same with rocks. Ken Hovind says things like, will this rock ever evolve into Mount Rushmore, for example? And of course, the blind watchmaker, would Mount Rushmore have come from a person or an intelligent design? Mount Rushmore never started off as a simple system. First of all, the reason the CDs will never turn into Windows XP is because the CDs do not self-replicate. The CDs do not create multiple offspring And from those offspring, just like dog breeding, we don't pick the one that has more of the trait that we want. So we pick this one and get rid of all the others. And now this one breeds into offspring. And again, which one is more like Windows XP, where it has code for DOS system or whatever? We pick that one out, throw the others out. And then again, we just keep doing this for generations. Self-replicating and variation in your offspring is an essential part of this change. That is why a rock will never turn into Mount Rushmore, and it's why a factory or a small mom and pop store will never turn into a factory, and they can't be compared. And if they do, because say a corporation like Amazon wants to build it up, it only demonstrates that systems get more complex through a term called emergent properties. So I wanted to talk about origins of life. At the beginning of life, life forms were very simple. They had very metabolisms, nothing like we have today.
I disagree. I will back it up with science.
Again, simple complexity is a vague term. I can at least agree on that part. Simply complex, it is a vague term, but comparatively simple. They were comparatively simple. However, they had traits that enabled them to survive a little better, and they got more complex. This is called an emergent property. It's the same reason why if you have two people working together, they can now lift up a box, whereas one person alone couldn't do it. And if you get three people in, not only can they lift the box, they can maybe assemble something together better. Then if you get four people together, well, now they can, have a party or something afterwards. And then you get 10 or 100 people together. Now they can form a factory where they not only put these together, they build these boxes themselves. An emergent property is the fundamental theme of biology, where things that get more complicated have more of an advantage and they are able to function differently. That's why your digestive system works with your circulatory and your respiratory system. That is all emergent properties. So I get the blind watchmaker argument. It's been used by creationists for a long time. And the arguments against it are it's not a comparable system to a biological system any more than Mount Rushmore comparing it to a rock.
It is. The genome is like the blueprint. The proteins are like the molecular machine.
Go ahead and put mine up.
Ribosome is like a factory inside of the cell factory, and the cell is a chemical factory in a literal sense. And if you want, I can show you many science papers.
But it isn't Amazon. It isn't Amazon. That is an analogy.
Todd, can I finish?
Yes.
And these science papers, they do not describe the cell as an analogy, as a factory, but in a literal sense as a chemical factory. So, I mean, you have to explain how the first cell emerged randomly, and you did not have evolution, because evolution starts with DNA replication.
But abiogenesis and evolution are not the same field. They're not.
Yes, that's what I am saying. But this is a problem for me, because you do not have evolution. You do not have evolution to explain the origin of biological cells. So if you don't have evolution, you have literally chance. So how do you want to explain the origin of life and cells by chance? That is the big question.
I can get into that question. At an hour and 13 minutes into this, I've been responding to your slideshows. I haven't put any slides up yet. So I'm going to put up here. And that's where I can, if you would like, show some things on, well, I don't know if I had pictures, but talk about the primordial Earth. So I would like to throw this out there, because this was something in my early days I was most baffled by: humans and primates and our descendants. And this is simple enough that everybody watching can also understand. So I'm hopefully not just throwing out a bunch of biochemical concepts and trying to overwhelm by default here. So hopefully everyone remembers from biology. On the left here, we have a human karyotype, which is our 23 chromosomes, and they are paired up together. You remember you get one chromosome from your mom and one chromosome from your dad. So you can see on the left there, that is, again, a human karyotype. On the right, we have a chimpanzee karyotype. They have 24 chromosomes for a total set of 48. So this was a formidable challenge, I will say. And I remember being a student in biology years ago. This was presented, and I should say 20 years ago. For us, it probably wasn't as long ago as other people, but all the great apes of which we are descended, all of these great apes, they have 24 chromosomes, and you get one from your mom and one from your dad, so that's a total of 48. So why do humans and our lineage, including Neanderthals, by the way, why do we have 23? that would seem to be a loss in information there. And that did pose a problem. Now, that doesn't mean automatically that therefore evolution is overturned. When I was younger, we actually speculated that one whole chromosome was lost. It just disappeared. But we knew that would probably cause problems. You are familiar, after all, with certain monosomy or trisomy diseases where, like Down syndrome, you get an extra chromosome, it can cause problems. And that is a small chromosome 21 as well. So we really struggled with this as a scientist for all these years. How can we have less chromosomes than the great apes if we have a common ancestry with them? And again, we did not evolve from chimpanzees. We and chimpanzees are cousins with a common ape-like ancestor. And by the way, we are still apes. So here is the thing that is most baffling to me. Because the answer did not come from lost chromosome, it turns out we were able to answer this in 2008. Now, 20 years ago, almost equivalent with putting men on the moon, was a grand project called the Human Genome Project. Very fundamental because we wanted to map out all 23 human chromosomes, all the genes, what are all the specific little traits. This was a 10 or 11-year undertaking to do this. And we thought it was going to take a much longer time. So it really was, and it was under budget too, which is another miracle. With the success of the Human Genome Project, at this point, I was already graduated and in college in that. We were starting up the Chimpanzee Genome Project and the Neanderthal Genome Project. And there's all these other genome projects, and they're all faster because we are better at it. But that's when a remarkable idea came out. This is the human karyotype on the left, and you can see the chimpanzee karyotype on the right. We've gotten rid of the pairs, so instead of one from mom and dad, you just have one chromosome there, unpaired, and you can see all 23 chromosomes. They look very similar, and now that we've mapped out the DNA, all of those loci, which are the specific gene segments, they are very similar to the chimpanzees. But again, where is the missing chromosome? And the answer came in a very bizarre way. Turns out it's chromosome number 2. But look at in the chimpanzee, their chromosome number 2 is nothing like ours. But there's the answer, 12 and 13. So if we take this here, notice there's 2 centromeres, by the way. These centromeres are the part of the chromosome that is in the middle part of the branch there. If you take the chimpanzee chromosome 12 and 13, and you flip it upside down like this, and we stack it on top of the other, just like this, watch what happens. We see a beautiful transition. All the loci suddenly match up. That was a very fundamental thing that came over 10 years ago, and it just blew our minds away because it really did not fit our predictions. We thought that there was a missing chromosome, but this still is a remarkable discovery, and it's even more cool. It means in our past, we had a common ancestor that had a fused chromosome, but evolution can make another prediction. Because if you can see here, I don't know if you can see my cursor when I wiggle my cursor or not.
Yes,
okay. There is a centromere here on the chimpanzee chromosome number 13 and a centromere here on number 12. The centromeres, for anybody watching, are just a protein-packed region in the chromosome. There's a specific motor there that attaches for mitosis when it divides. So the idea is, well, where is our centromere in chromosome 2? If it has a common ancestry, we should find this kinetochore protein as a fossil. It took a few years. We found it. There is a motor protein. It is a fossil partial remain of a centromere exactly on the right loci for chimpanzee chromosome 12 and 13. And when you look again at the loci, they are not perfect matches. They are very similar. And the more crazy part about this is if you put a horse and a donkey chromosome next to each other, chimpanzee and human chromosomes are much more similar than a horse and a donkey chromosome. And yet horses and donkeys, we always say, if you look at them, we always say they're very similar. They can even mate and produce mules. Now, I'm not going to speculate about humans and chimpanzees. We could never produce an offspring with them because our chromosomes do not match up. They wouldn't. You couldn't have a 23 and 24 chromosome match up homologously. But again, if you look at chromosome 12 and 13, we have every bit of DNA more in common with them than a horse and a donkey. This was a prediction that evolution made, but it was discovered in 2008. I had already graduated, I had already learned, and I was blown away by this. And I have to say, Young-Earth creationism has no explanation for this. Not even Young-Earth, but anybody who is saying that this is all divinely inspired, it's just saying God did it. It is not an explanation. But there it is. It's in our DNA. So there's my thing to you. Do you have a thought for that? And again, I know you're not a geneticist. If you do not have an answer, that is okay. But that is a presentation I can give you on that.
No, I have already put on the link. It's gone. Let's wait for Todd to come back.
Can you still hear me? Hear me? Okay.
Yeah.
I went to close the screen, and what ended up happening was I clicked the button where it says Leave Studio. So again, to anyone watching, he's using a software called StreamYard. This is my first time, so I'm an amateur at this. Anyways, just wanted to throw that up.
So I have already put a link on the chat as an answer to your point, Todd. This is a very, very well-known talking point of proponents of evolution. It's nothing new, which are saying, okay.
It's pretty new, 12 years.
Well, whatever. I am not in the situation to give you now verbally an answer to that, but I have studied it. I have a link on that, and if you want, you can look it up.
I respect that you don't have an answer because I know you're not a geneticist. I share it simply because it is easy enough to understand, and it's not something I will say, because I have seen explanations that does have a good explanation. In fact, it was used in the Dover case too. I got another one for you. Vestigial organs.
You said that you are a biologist, is that correct?
Biology is in my background. I'm not a biologist with publications or anything.
Okay.
Okay, I'm going to throw another one out at you here, and I'm probably going to keep my screen here. Everybody knows about vestigial organs. Now, one common misconception I heard all the time when I was a creationist was, why would we have these? They must have a function. And so a lot of times creationists will try to say they have a function. That is not what a vestigial organ is. A vestigial organ, by definition, is something that is a fossil remnant of our ancestors. It doesn't have the original intended function, but it still might have a function to it. So the appendix is one that is commonly used, and it's commonly mocked, but there's many more out there. But let's just talk about the appendix for a minute. A lot of times we look at the appendix, it causes problems, appendicitis. In fact, my father-in-law almost died of appendicitis. But the argument from creationists is, well, slow down a minute. God did it for a reason. Again, God did it. There must be an answer to it, a function to it. So one thing commonly cited is that it could be a pouch for bacteria. And if your gut has bad bacteria, it can clear itself out. And so having that little reservoir in the bottom with that bacteria, that actually can maybe repopulate your gut. That hypothesis has been looked into, and it's also been suggested that embryos or developing fetuses may also use it for that purpose. Okay, I have no problem with that. And in studying evolution, it is not really a problem with most other people too, because it may serve as one of these. After all, it is on our body, so our body may even try to find another function to it. So the appendix, however, we need to look at what is its original function, and where do you find it? You find it in herbaceous animals. These are the different animals here. You can look at different examples here, marsupials and whatnot, and not to mention, well, lagomorphs, the rabbits and that. These are animals that eat herbaceous matter. The appendix is there because it serves as a pouch for digesting cellulose. Now, you and I, we obviously have the ability to eat meat and we have the ability to eat plants, but our ancestry tells us a story that we were plant eaters. It's even in our teeth. We are from plant-eating animals that later adapted to eat meat. And we can eat meat, but we do struggle to eat it more than, say, your dog does. We digest it a little bit less efficiently than them because we are a jerry-rigged herbaceous animal, and we still have this part in us for us today. But another one, what about goosebumps? This is one I've never heard an explanation for from the creation crowd, because we can talk about the appendix and, you know, it may or may not have another function. But no explanation I've heard of, and I don't expect you to have one here either, because again, this is on the spot, but no explanation has been given for goosebumps. Goosebumps are a genetic trait we have, and they are for the purpose of fluffing your hair up. But they also fluff your hair up to make you look big and scary when you are scared, so you can defend yourself. This is an essential part for many animals. Why would we have these, and why are they on our hair follicles, and why is it the same genes that do this that is in the other animals? Again, creation has no explanation for this. And again, evolution predictions, they might be wrong too. This is, after all, a theory. It might be wrong, but the evidence seems to fit with the idea of common descent, that you and I have goosebumps because our ancestors had more body hair. In fact, you and I still have body hair, and I will throw this out there. You have the same amount of hair as a chimpanzee. Yes, you do. You just have thinner hair. So you might notice women have even more thin hair than us, burly, hairy men have. But women have the same amount of hair we have. It's either just small or underdeveloped. And chimpanzees just have a really coarse hair. We still have that body hair. We just don't have use for it because it isn't very fluffy on us. But that's why when you get scared in that. The tailbone is another one brought up there. It doesn't really have any.
Would you like to talk as well? Because as I said previously, you're taking about 80% of your time to talk. Even the people in the chat, they are complaining about that.
I don't really care because I'm sure this is pretty one-sided. I really don't. But if anyone has a question, go for it.
Well,
I just want to spend after an hour and 12 minutes talking about irreducible complexity. And like I said, I don't expect you to have answers to these. That's why I'm not trying to read off long paragraphs here. I'm just trying to throw this out there and try to give a little more insight onto this. But
as a general critique point, I would say that in order to elucidate both evolution and abiogenesis, where the action happens, it is in biochemistry. And as you said that you have no knowledge in biochemistry, then I would suggest before that you try to teach others about evolution, learn about biochemistry, because there is where the action happens. And that's where the arguments are either lost or won. Okay?
I'm smelling a little deflection right now. I've already said that, and I'm honest about that. And by the way, I do know some things about biochemistry. That is part of having a bio background. But on this topic here, if you have a thought, otherwise I'd like to share the other things here.
Well, how many things would you like to share? Because
you read whole paragraphs off to me on Tango. I'm not reading paragraphs off to you.
Well, I have merely a chance to give an input. You are just talking. You are not listening. And I mean, it is a little bit one-sided in my understanding, Todd.
Look at the chat box. I hardly would say that's one-sided.
Okay, I'll give you more 2 examples, and then I think we have already 1.5 hours.
I'm not upset. I can stay on. But again, I'm trying to show some examples here, like the tailbone. What about the nictitating membrane of your eye? That's still there. You can pull back on it. I can throw another one about the jaw. Herbaceous animals need a longer jaw for that. What about the palmaris longus muscle that's in your wrist? This is used for things with long fingers. It has no function, and 15% of people do not have this. But if you have long fingers, say the great apes, this muscle has a function for us. Darwin's ear tubercle, I'm not quite sure the function, but a bunch of us do not have it. I do have this little bump there. It is a remnant, okay? Muscles of the ear. You can come up behind somebody. If anybody in the chat has a little sibling or something, run up behind them and blow a little whistle. Now, they can't probably move their ears, but their ear will turn to a high-pitched sound. Why would you have that ability to do that? You cannot hear sounds over 17,000 hertz, but yet your ears will respond, okay? Now, we could talk about hind legs. Creationists love to mock this idea here, but again, what is the purpose? They were land animals that evolved back into the water. Muscles still attached to those, yes, and snake may use those legs for mating, but again, vestigial organs does not mean it can't have a function, but they came from those original genes. And why in the world do manatees have fingernails? Come on here. This is a land animal. It was a land animal, and it evolved back into the water as an aquatic animal. That's why it has this. It makes sense in the light of evolution. And again, creation cannot make a prediction of this. We can also say flightless birds. Penguins have flippers. Now, these are not for flight, but why would they have these? And the answer is because their ancestors used them for flight. They have readapted for another purpose, but that is not the best way to do this. Either way, what about the emu or the ostrich? They have a dew claw that doesn't really do anything. It literally just hangs there like this, a little dew claw, and they can fluff their chest up. But why do they have these arm bones still there? And again, I would apply that same logic to Tyrannosaurus Rex too, by the way. It didn't necessarily have to have a function on its arms. Now we can talk about embryologic structure and move it here, but that's a whole nother idea. I will leave it to, and I can get on that in a minute, but again, my claim here is vestigial structures. And so why, again, is that a problem? How does creation, if I had a chance to make a question, make a prediction that can be answered by vestigial structures?
Well, as I said, I think that your entire approach is inadequate because the mechanisms which define organismal structure and body plan are on the molecular level. And if you do not have knowledge of what...
That is where natural selection takes place too, by the way.
Well, that is your assertion, which I completely disagree with. And I can give you...
No, not at all.
I can give you afterwards a link to my investigation in regards what the real mechanisms are which define organismal complexity and body plans. And it's not gene-centric. It has to do with epigenetic languages and codes and signaling pathways, and they have to be pre-programmed. So I dismiss your entire approach because it is not where the action is.
But again, you're saying, Antangelo, that these are so complex, God must have done it. That is basically the underlying argument here. And so the problem is that is a negative argument.
My argument, Todd, is that what we require in order to make complex multicellular organisms is pre-programmed information in genes and in epigenetic languages. And I have collected over 30 different epigenetic codes which are involved in organismal structure and development, and also signaling pathways. And these languages, they have to be established first in order to function. And this is undoubtedly something that only intelligence can do.
Okay, so again, the reason I do not have my son's first grade teacher work on my car is because that's not her background. If you have all this ideas and stuff, the proper approach would be to publish this and go out there instead of sharing this in inner circles.
Oh, it has already been published because my inference is based on my inference is based on peer-reviewed science papers. After our chat, I can provide you the links. I have tons of peer-reviewed science papers which are the basis of my inference. So I do not have to publish anything because it's already out there.
But I will assert here that everything that is based on the argument of irreducible complexity fails when it is put under the test of peer review. And that's why spending time just going over biomolecules and that and saying, I can't explain how this happened, God must have done it. There's no natural explanation.
You've not seen me anytime. You're strawmanning what I am saying, Todd. I've not said that we don't know how things happen, therefore God. I said that what we see...
Is it intelligent to design the inclusion?
Can I finish? You are not letting me finish, okay? What I am saying, and I repeat myself, I will actually put it on the screen so you can have a look at it, because I'm investigating this for years, and I know what I'm talking about, okay? So just give me a minute, and then I will put it on the screen, and you can have a look. And when we finish our chat, then I will provide you the link, and then you can look it up. But I entirely disagree with you that genes alone define organismal complexity and body form. That is not what is. That is not the information which is provided by current science. Okay.
I'm sorry. Are you putting something on the screen?
Yes, just a moment.
Okay, go ahead.
Okay. I am listing here 19 different mechanisms, and genes are a very small part of this. And I can provide you afterwards all the links to the science papers which back up this list. All these 19 mechanisms, they are involved in organismal development and organismal form, and they define also the structure of each cell, what kind of cell that it will be, what the organismal development, all this kind of stuff. And it's not gene-centric. The overarching explanation of all this is here. Blueprints, instructional information, and master plans which permit the striking autonomous self-organization and control of complex machines, molecular machines, and exquisite factory park cells. Upon these are both always tracked back to an intelligent source which made both for purpose-specific goals. And that brings us unambiguously to intelligent design. Let me just end here.
Okay, I did it. That's the argument again.
To explain organismal complexity is pre-programmed instructional complex information encoded in various languages and communication through various signaling networks that act on the structural level, which are pre-instructed to respond to environmental cues, development, and nutrition demands. This is basically the entire difference, which in my understanding replaces evolutionary biology and the gene-centric view of natural selection, of mutations, of gene flow, and so forth. So what we see is that the information has to be first pre-programmed, and you need to establish the languages, the gene language, the genetic code, and also epigenetic languages. And there are almost 30 different epigenetic codes in place. I am listing them here.
Okay, but again, that is...
All these 29 different epigenetic codes, they are involved in organismal in body plan and
yes.
building. So you have to ask yourself, can codified information and codified languages originate by unguided random evolutionary mechanisms? And this is the big question.
Okay, but yes, and to answer that question, again, these examples are with modern organisms, because you're right, you cannot remove these from our DNA and have it function. These are necessary epigenetic codes in there. But cells are not always the same like this. We're looking at modern organisms here. In order to make a life form, something like you said, it has to be able to replicate. RNA can self-replicate. RNA can polymerize, and those mononucleotides can form naturally. Now, that doesn't mean that they're going to have a gene. Well, here, I was trying to answer this, and this is what happens. And I know it's a lot, but again, it's like when RNA can self-replicate, that doesn't mean it's going to make life right away. And no, it's not going to have all the codons in place to shoot out one code that's going to make this. But inside, and we didn't even get into the primordial concepts here, but inside a protocell, the micelle, the cell membrane, which provides an environment outside of the exterior, that protein or that RNA is more stable from the outside environment. That's going to give it an emergent property. Now, if it makes copies and then the cell divides by binary fission, these, by the way, have already been done in the lab. We now have another emergent property. They can self-replicate and they can binary divide into two. Once that happens, all it is just a matter of modification. That doesn't mean it's got an end goal in mind. It's not trying to make these, but the RNA will continue to get more complicated. So I forget the names of the scientists, but in 2014, this was an experiment they did where they took RNA with the different chiral shapes, the left and right-handed of it, which were originally a concern because when we did abiotic experiments with them, where we could see if they got created, they were said to not be able to coexist, but they created conditions that were just right. And not only were they able to replicate, they got more complicated. Those RNA since 2014 are continuously dividing and becoming increasingly complicated. Now, it didn't make a cell and nobody is expecting it to make it soon, but all you need is self-replication and change. That means variation in the offspring. When that happens and you have those two conditions, it is inevitable you will get more complicated emergence. So no, I do not have the biochemical answer as to those genes, but we're looking at today, you're looking at the top of the hill and saying, how did you get from the bottom to the top? And the answer is not chance that these all fell into place. The answer is emergence, that little things here stayed. They were able to self-replicate. We can do this in the lab. It's called protobionts. They do self-replicate with RNA in them. When they self-replicate, they get more complicated. And again, this is a postulation that over time, we're talking almost 4 billion years now, you have a divergence. But keep in mind, in the theories of life that we have, 3/4 of Earth's history was just small protobiotic organisms. Only the vast majority of the phyla we see today were emerged in the last 500 million years, a very small percentage of Earth's history. Life, it is hard, but it diverges very quickly once it gets complicated. Anyways, I see another slide here.
Yes. What would happen if you would have RNA on a prebiotic Earth? And this has been, this is a peer-reviewed paper which has been published in 2012. I can give you the link afterwards. Stephen Banner is a very well-known abiogenesis.
I know Stephen Banner.
And he says the systems given energy and left to themselves devolve to give useless complex mixtures, asphalts. The literature reports to our knowledge exactly 0 confirmed observations where replication involving replicable imperfections, evolution emerged spontaneously from a devolving chemical system. It is impossible for any non-living chemical system to escape evolution to enter into the Darwinian world of the living. Such statements of impossibility apply even to macromolecules, not assumed to be necessary for really evolution. So that is what would happen on the prebiotic Earth. These molecules, they would be burned by UV radiation and simply decompose. They would not complexify and self-replicate.
Okay, hang on. So Darwin said the same thing, except he didn't know the full things. He published a letter back in 1860 or 70 or something, and it was the idea that surely, because he was just speculating, he didn't have anything, and he said, whatever early life may have formed from, surely it would have been simple, but at the same time, the conditions of the Earth surely would have destroyed them. And RNA, you're right, it is not stable when we leave it out. It tends to degrade very quickly, and you're right, UV light is one example. And to your credit, the early Earth would have had even more UV light because there was no photosynthesis around. Without photosynthesis, you don't have ozone. And so, yeah, there would have been a lot more UV light that would have been around. And so that would have been a problem. However, UV light and the conditions, volcanism, not to mention there's a word for all the impacts that we had back then because there was a lot more space debris, asteroid impacts. In fact, most of the moon was formed, or not the moon, but the face of the moon we see now was formed at that time from all those impacts. If we go back to that period in time, RNA, it was able to be created. Now, it wasn't a complex code, but the mononucleotides were formed. And this is what I was saying earlier. We have seen this in a lab. Now, again, when they polymerize, they do tend to get destroyed very quickly. However, this is where emergent properties comes into play to answer this, because RNA, it does get destroyed very quickly by UV light. But if it was inside of, say, a membrane or something, now it has a little more protection than it did. It might not be perfect, but a membrane, and by the way, those lipid membranes that form the cell membrane, those naturally form too. Suppose one of them got stuck inside of it. Now, we have RNA that retains the ability to self-replicate. That would have been chance too, by the way. But again, this is not evolution. This is abiogenesis. They would have had to have got stuck inside of the protocell, inside that membrane. They retain the ability to copy. And being inside of that membrane, now they have another property, and that is slightly, not a lot, but slightly more UV light protection. Now, suppose that they also formed in a specific environment. This is the work of Sidney Fox in the 1960s. Actually, his first work was in 1958, where he tried different environments with these RNA molecules and proteins. And he found one place, believe it or not, was actually on clay substrates. He had to dry it out, wet, dry, wet, dry. we wouldn't have thought those would have been an ideal condition. But polymerization happened, and inside of a protocell, they were protected from UV light. So yes, he is right. Those would have been destroyed. But in certain conditions, protected by a membrane, they have an extra shield against UV light. And not to mention, there would have been a lot of vapor at the time too at this. And this also would have served as somewhat of a protector. But it only would have been certain places on Earth. The entire Earth would not have been protected from UV light. The deep parts of the water also would have been protected, but not the shallow parts. So there's a lot of different things to this. We do not know, but as scientists, we make a hypothesis and we test it out. Now, bringing up his quote there, by using it, it could be seen as a potential appeal to an authority there, because the truth is all of us who have ideas, we can be fallible. So we subject this to peer review and reliable sources. My best guess from what he is saying here is that these are not, my best explanation here for what he is saying is science is not showing that. These can form even in those harsh conditions, but that is left up to chance. Once they self-replicate, though, and they're able to divide by binary fission, it's pretty much inevitable at that point they will get more complex with more emergent properties.
Okay, a paper which is much more recent from 2006 from Robert Shapiro. He says the natural clays catalyze this reaction. The attractiveness of this oligonucleotide synthesis rests in part in the ready availability of the catalyst. Montmorillonite is a layered clay mineral rich in silicate and aluminum oxide bonds. It is widely distributed in deposits on the contemporary Earth. If the polymerization of RNA subunits was a common property of this native mineral, the case of RNA at the start of life would be greatly enhanced. However, the catalytic activity of native Montmorillonite before being converted to the homoionic forms is very poor. The native clays contain bound polyvalent cations such as CSO and so that interfere with phosphorylation reactions. This handicap was overcome in the synthetic experiments by titrating the clays to a monoionic form, generally sodium, before they were used. Even after this step, the activity of the Montmorillonite depends strongly on the physical source, with samples from Wyoming yielding the best results. Eventually, the experimenter settled on wall clay, a commercially processed Wyoming Montmorillonite provided by the American Colloid Company. Further purification steps were applied to obtain the catalyst used for the prebiotic formation of RNA. Of course, you didn't have purified Montmorillonite on prebiotic Earth. These experiments, they are highly tuned in order to work. So you can forget that as an example, which proves that polymerization of RNA could have happened on a prebiotic Earth.
Okay, I'm not really sure how I'm going to respond to that, because I'll be honest, it's small writing on my screen, so I was mostly trying to listen to that.
I can.
It looked like it was on a message board, and it was.
I sent the link after our chat.
And I hate to say this again, but I'm just going to throw this out there, because other than my PowerPoint, I've basically been going by, and I see everyone in the chat seems to say all I'm doing is talking over and that, but I have only had a chance to put up my PowerPoint that one time. I've been the whole time listening to slides being read, big paragraphs, and that last one was just a little bit hard for me to read because my eyes are hurting. And I don't know what time zone you're in, but it is approaching 11 here. And I can certainly carry on, but if it's just reading little things like that, it's going to be hard for me to follow and respond to, just unfortunately.
Yeah,
I would say, first of all, if we were doing a scientific investigation, a message board response like that is probably not a source I would use to say as a baseline. I did note it was from June of 2006 as well. And I also can say that we've learned a mountain of things in the last 14 years. Heck, at that point, we didn't even know about the chimpanzee chromosome with humans. And so I can only imagine how much more they have learned since that time when it was published. I will throw one thing out here really quick too, since this, let me throw one thing here too myself. So to someone in the chat who was just, I wanted to throw this out here in the chat real quick. This person has a genetic disease. And to everyone in the chat looking here right now, this person has a genetic disease on their fingertips. So how do you suppose that genetic disease got there? And of course, it goes back into the biochemical pathway. Except, wait a minute, this isn't a person. This is a gorilla finger. this gorilla actually has pigments that are not forming. You look at that. That is a human finger. That is an ancestor. Well, not an ancestor. That is a cousin of ours. There is evidence in evolution in our body structures. And so we can look at things like this. And again, I agree, it's in the genetic code. This is a gorilla, for Pete's sake. We can look in our embryos, in our development, and we cannot misrepresent this. We have similarity. How can you not look at things like that and see a similarity? It's kind of like when I see people that on TV or Maury or whatnot, they get a paternity test and they see the DNA evidence and they say, that's not my son. And the mom is sitting there saying, he looks like you. And she's yelling back and forth, he looks like you. And the guy keeps sitting there and says, that's not my son. I will say this, and then I will probably bid you adieu here. You're looking at that gorilla picture. You know there's a resemblance. They have fingernails, forward-facing eyes. They have the same amount of hair as density as us. Chimpanzees, by the way, are more similar to us than chimpanzees are to gorillas. They might look different. They might look hairy and we look different. But in our genetic structure, and when you look at our comparative anatomy, we have more in common with a chimpanzee and a closer common ancestor as a cousin than they do with gorillas. And yet, even looking at that gorilla finger there, that looks just like a human. So I say to a creationist on here, look at this. This does not mean God could not have been involved in one way, but this does mean that you have to face the evidence here, that this is not just something we can simply wash off as God did it, and there's no explanation behind this. God just made it that way because he was creative, because we can make predictions behind these. All right, go ahead.
Okay, Todd, you have here on the right side the eye of a single-celled organism. And on the right side, you have the example of a human eye. And they are convergent. They are similarly structured. Okay? So how do you explain this? They are completely different organisms with convergent biological systems. Yeah.
Okay. That would be they were independently evolved. In fact, the eyeball has evolved about 40 different times. In fact, when you look at spiders and insects, we all think of them as bugs, but they're actually the arachnids. They are not even closely related to the bugs, except they are arthropods. They have an eye shared all in common with the other arthropods, scorpions, ticks, daddy longlegs, if you want to call them, the harvest men and all of that. They all share the same eye. But then when you look at the insects, the 6 legs, 3 body segments, they have the compound eye. It was independently evolved. Then when you look at the octopus, the squid, all of the mollusks that are those type have the same eye. Our eyeball is a vertebrate eyeball. And we all share the same genes that make the lenses, that make all the parts. And if you look at our eye and a bird eye and a reptile eye, they have those commonalities. Now, why won't you ever see a reptile with a compound eye? Because they didn't evolve from an insect. They never got it. Vertebrates independently evolved their eyeballs, and anybody who is a descendant of you will have that as well. Why do Europeans have blue eyes, or some Europeans, I should say? Why is it not scattered throughout the world? And it's because thousands of years ago, somebody was born with a mutation. They had a blue eye, and anybody today with blue eyes is a descendant of that first person. If another person mutated in, say, Japan and had blue eyes, and say it was in a different place or whatnot, maybe a different pigment form or whatnot that gave them a blue eye, well, it wouldn't be from the same path. It would be blue eyes, but it would be scattered amongst their descendants. And that's why we would call it convergence, because they look the same, but it did not evolve from the same common ancestor. You and I have legs, and so does a spider, but they did not come from the same common ancestor. Spiders got their legs independent. And one more thought. We can use this same prediction. I personally don't know how I feel about aliens, but if the eyeball independently evolved 40 times and all of those phyla have their eyes independently and whatnot, classes and orders, and legs is a common theme, we could probably, and chemical sensors like vertebrates, we have noses, and insects have their antenna and whatnot. We could probably right now make a prediction that if we found alien life forms, that they would probably not have wheels. They'd probably have legs too, and eyeballs, because it's a common recurring theme that seems to be a preferred means of body function. We do not know why, but this seems to be, again, the preferred body function. And whoops, actually that brings up a cool, you've heard of the thylacine before?
What?
The thylacine?
No.
Here, I'll get it up here. It's called the, well, some call it the Tasmanian tiger. But I was asked by this by somebody, hopefully you can see this. I don't know if is it popping up?
Yeah.
Oh, okay, it's not showing on my, oh, there it is. I show this because I was asked this one time and they were saying, why do they, somebody asked me this on that creationist site. Why do people say that these are, we call them tigers, and why do evolutionists say this is convergent? Well, the reason why is they look the same. They look pretty similar. In fact, the Tasmanian tiger looks kind of like a dog, more than it does a tiger. We just call it that. But look closer. It's not a cat at all. In fact, it's not even a carnivore. It's a marsupial. That is not even closely related to the rest of us. We are much more closely related to that tiger than we are to the marsupial. Now, it is extinct, so this is a color photo from 1937 or whenever it went extinct, but we can make another prediction. You don't know, you just said nothing about this here, so that gives us a good blank slate. This creature here, it looks just like a carnivore, like a dog or maybe a cat. And so why don't we just group it together like creationists would do with baraminology, or baraminology as they call it? Because evolution allows us to make a prediction. If this is a marsupial, it should have the same traits, just like what I was talking about with the eyes. And if you look at it, it has a freaking pouch. It has little lactating glands that resemble little teats for the babies to crawl in, just like on the kangaroo, into the pouch. Why in the world would this have this? And also, look at the teeth and the mouth and the fingers. Cats have a claw that's way up, as you know, because they walk on their outer digits. But the tiger, the Tasmanian tiger, the thylacine, it has 5 fingers it walks on just like this. It has teeth, not like any carnivore. And look at its face. That looks just like a koala nose, not a cat nose like you see with cats and dogs and bears and otters where they have the whiskers and that. This animal has none of that. This animal is a marsupial modified with a body like this that just resembles a carnivore. But it in every way is related to an opossum and related to a koala and a kangaroo because it is a marsupial. It's just jerry-rigged with this body form. So why does it look like this? And that, again, what I was saying with aliens, it probably has there's probably something in development where being a carnivore prefers a sleek body type like this because you're fast and you can do this. And likewise, having eyes, there's probably a preferred path in evolution that it took with eyes to make. However, they do look a little different throughout. So, I mean, there's a lot of stuff to talk about with this, but again, at face value, why do I have to keep explaining this to creationists? This is very much a marsupial. It just looks like a tiger, but it's not a tiger.
I am not disagreeing with you, Todd. Anyway, John, good night. What's up?
Oh, and one more thing too. Sorry, because one more thing too. And this is fascinating, and that's why I don't want to forget about this. So I really don't want to interrupt here. But I was at the zoo a few years ago, and I saw a kangaroo hopping, and it was a male kangaroo because you're always trying to see what are the boys and girls, you know, and you see this male kangaroo hopping and it has, you know, you can clearly see the penis and all that below it. And then I saw this tumor hanging off of its stomach. And I remember thinking, oh my gosh, like this thing probably needs to get checked out. And it was a questionable zoo. So I thought maybe this zoo needs to like be investigated. Maybe this is a tumor hanging off of its belly. And then I noticed another kangaroo, a male with the penis and that, you know, but it had another tumor hanging off of its belly. And I was very disturbed by seeing this. And I was like, what in the world is going on with these animals? Well, then I went home and I started looking into this. It turns out the kangaroo, the scrotum does not go behind the penis. It comes out the belly. And that's because as mammals, our testicles, maybe you know this, they develop up below our chest. They develop as an embryo and they descend downward as your mother is pregnant with you and you're in utero. They go down in between the legs and they pop out in the scrotum. However, we still have the same blood vessels that fish have all the way up by where our rib cage is and our chest is at. Okay. Now, having said that, I went and could make a prediction with evolution. Clearly, the kangaroo, when it was developing, the testicles in these marsupials, it went down, but instead of popping out between the legs, it popped out in the stomach and herniated, if you will. basically making a scrotum out of the tissue from the abdominal wall. So I remember thinking to myself, and I was talking to my wife, like, here I was accusing the zoo of, letting their animals get cancerous. And then I thought to myself, I bet you koalas have that too. I bet you everything else that is a marsupial probably has that too. And I got curious, and this was probably the weirdest thing I've ever had to search for. Sure enough, male koalas have this weird scrotum-like structure hanging off of their abdominal wall, not below the penis. Now, again, it doesn't prove evolution. You cannot prove this. Why would all these marsupials have this in common? Creation cannot explain this. It makes no prediction that can be tested that could have explained that. But I was able to make that prediction before looking it up, because evolutionary theory has been tested for 150 years and it has withstood the test of time. So yes, biochemical and all, it's shared in their DNA, but it is there plain as day. And I will leave you with this thought here. Again, you are looking at evidence. You can be like the father who sees the DNA test and denies that is your child, but look at the evidence before you. This is here evidence of common descent with modification. And anybody who is related to those things will share those features. But if you are not related, you will only resemble them if you converge and maybe have a similar niche, maybe chasing prey or something. But you will never be a cat. You may look like one, but you will never have the paws of a cat. You will never have the whiskers like a cat or the nose, because you're not a cat. You're just a thylacine. Okay?
Well,
all right.
We agree with limited common descent, and this is explained by speciation and macroevolution of second degree. But common descent of all life forms, this is something which I strongly disagree. I have evidence that this isn't possible. And also macroevolution of first degree of speciation thought, as I told you, in order to explain how organisms are formed, you need to look into biochemistry. And
biochemistry shows methods of mutation and natural selection, which explains the mechanism. Darwin would love to have seen the day Watson.
I strongly disagree for the reasons which I brought up previously. I showed you that over 30 epigenetic languages and codes are involved in organismal building and also signaling pathways.
And sir, like I said, those are today, but they are not needed for life. I did not agree with your definition of life, that complexity in its current form is needed. No biologist would, because we are still defining what is life. Viruses are not alive, but they are biological systems.
Todd, I didn't claim that these epigenetic mechanisms are required to explain the origin of life. Nonetheless, they are required to explain the origin and development of complex multicellular eukaryotic organisms. So
again, that is irreducible complexity.
Todd, do you actually know anything about genetics? Because you keep going on these tangents and claiming that Otangelo doesn't know anything.
Answer the question here, and I mean, really, that is your question. Irreducible. I mean, with irreducible complexity in genetics.
My point was not about irreducible complexity, Todd. I was talking that there are over 30 epigenetic languages and codes involved in organismal building and development of organismal form, and that has nothing to do with irreducible complexity. Now, in order to explain the change from one kind of organism to another, you have to put into the toolbox also these epigenetic languages which are involved in development.
Those were not in the original organisms. They're not even in many viruses. Viruses don't have them, but today we are only alive because we all survived the Permian extinction. 99% of life went extinct at that point. We were bottlenecked.
These epigenetic languages cannot be explained. Todd, these epigenetic languages cannot be explained by genetic variation because they are. Can I finish, please? Because they are inherited epigenetically. There is no genetics involved in inheritance of these languages. There are many things like also cell memory, which is not inherited by genetics. So you cannot invoke evolutionary traditionally gene-centric mechanisms to explain the phenomena.
That same argument was used years ago when they said altruism could not be inherited and it was not possible. Altruism, for anybody listening, is when you do things for other people that benefit them, but not yourself. In fact, it may even make your life more at risk. And that has been argued very commonly in creationist circles, where they say you could not care about your fellow man because evolution is all about caring about your own survival.
Todd, what the heck does that have to do with genetics, biochemistry?
It has to do with
no. Stop interrupting everybody, bro. I've been listening to you guys for an hour and a half now, and you've been talking over Otangelo this entire time. Actually, let people finish concepts before you spew your talking points, most of which have already been debunked. And then claiming when you don't know what the heck you're talking about, you then claim, oh, I'm not an expert, but whatever point you're making is wrong.